Thank you very much for inviting me to speak. It's a great pleasure and honor to be here. I'm very excited to tell you today about the work that we are doing, and please feel free to tweet any parts or slides of this talk. So I'm going to tell you about a problem that has fascinated me for many years, which is how does a single cell form an entire organism, and specifically a human being made of thousands of single cells. And it's really important we understand that process, because failure to establish cellular identity early in development or to maintain cell identity in the adult results in diseases, but we still do not understand these processes fully. So scientists have been trying to explain embryo development for a very long time. Aristotle was already busy with this. He coined the term epigenesis, which is the process by which a single cell makes an entire organism. And so now we know, of course, that cells are gradually specified during development, gradually acquire cellular identity to end up with all the cells that make up the human body. And when I started my PhD in Cambridge a number of years ago, I met John Gurdon, who said that he could even reverse development and take the nucleus of a differentiated cell, introduce it into an egg whose own nucleus has been removed, and derive a cloned embryo now made of thousands of different cells that are genetically identical from the donor nucleus. And so these experiments really mean that you can go from one genotype to acquire multiple different phenotypes and cellular identities during development and reprogramming. And so how is it that one nucleus can adopt so many different cell states during development and reprogramming? Well, we know now that this comes down to gene expression programs, and that different cells express different sets of genes. And this comes down to having the right activating proteins on chromatin, activating the right genes in the right cells, and also repressive chromatin regulatory proteins that inactivate alternative developmental programs. And so it's really about how gene regulatory programs and chromatin regulate development and maintain cellular identity. And so the key question that I want to tell you about today is, how do gene regulatory programs and chromatin regulate early mammalian development and reprogramming? And we've made tremendous progress in understanding this over the last decades, but there are still many questions that remain. So what I'm going to do is I'm going to take you to three exciting pieces of work. The first is on chromatin control of lineage induction early in mammalian embryogenesis. The second is on chromatin and dosage compensation in mammals. And the third is on post-implantation human development, where I will tell you that we have identified a way to isolate the missing cell type. So we are very interested in understanding chromatin regulation in the context of mammalian development, and specifically in the context of early human development. And so if we look at the human embryo, one of the first things that happens is that the other trophectoderm cells are specified, and these trophectoderm cells then give rise to the entire trophoblast lineage, which basically makes the placenta, which is key for embryo growth. And then the inner cell mass of the embryo then undergoes second cell-fate decision that segregates naive epiblast cells from primitive endoderm. The naive epiblast will give rise to all epiblast cells, and that will give rise to the entire embryo, the entire all cells of the body, while primitive endoderm gives rise to the yolk sac. And so establishing these three founding lineages is really critical. It's gradual during development, so cell-fate decisions take place sequentially, or in pre-implantation and thereafter as well. And there is also formation of a cavity, morphogenesis, that is key for implantation and downstream events. So if we look at the three founding lineages of the human blastocyst, the trophectoderm, naive epiblast, and primitive endoderm, the embryo looks remarkably similar in mouse and human, but there are very big differences. In the mouse, all three founding lineages are fully restricted by the time the embryo implants, meaning that cells are irreversibly committed. They cannot switch from one lineage to the other. However, that's not the case in humans. In humans, the blastocyst just before implantation is still plastic, meaning that trophectoderm cells can still give rise to the naive epiblast, and naive epiblast cells can also give rise to the trophectoderm in vitro. So there is a tremendous plasticity in the human compared to the mouse. We can isolate pluripotent stem cells from embryos and by reprogramming approaches as well, thanks to the work of Shinya Yamanaka. So naive human pluripotent stem cells can be isolated from human pre-implantation embryos, and they represent the pre-implantation state, meaning that we can model human pre-implantation development with naive pluripotent stem cells. And then conventional prime pluripotent stem cells, on the other hand, represent post-implantation epiblast. And I'm sure you've been told about this because they are a really great model for gastrulation in downstream events. And then there are formative pluripotent stem cells that are intermediate between naive and prime. So pluripotent stem cells offer fantastic tools to model early human development and ask how do gene regulatory programs and chromatin regulate early human embryogenesis. And in the case of naive human pluripotent stem cells, one of the remarkable property of these cells is that they can self-renew indefinitely, and they maintain the capacity to give rise to prime cells and the three germ layers, ectoderm, mesoderm, and endoderm. But naive human pluripotent stem cells have complete lineage potential, meaning that they can not only give rise to prime cells, they can also give rise to the trophoblast and primitive endoderm lineages. So this is a remarkable cell type that can basically make everything. And this complete lineage potential also enables naive pluripotent stem cells to form blastoids that are basically naive pluripotent stem cells that self-organize in 3D into structures that specify the trophectoderm lineage and then acquires the morphology of a human blastocyst. And so human blastoids model not only lineage specification following self-organization, but also morphogenesis, and can also mimic several aspects of implantation and model early post-implantation. So this is a fantastic tool that we now have to study early human development. But because naive cells basically can give rise to all lineages, they have been proposed to be epigenetically unrestricted, devoid of chromatin barriers that oppose alternative lineage inductions. And so the question that we wanted to address is, are human naive pluripotent stem cells epigenetically unrestricted? And what I'm going to tell you today is that we found that unlike previously thought, naive human pluripotent stem cells are not epigenetically unrestricted. Instead, they possess chromatin mechanisms that oppose lineage induction towards other lineages. So we wanted to understand how human naive pluripotency is regulated. And so together with Peter Regan, Hendrik Marx, Martin Danens, Michiel Vermeulen, and Nicolas Rivon, we have comprehensively identified the proteins that are enriched on chromatin in human naive and prime pluripotent stem cells. And we then wanted to know how chromatin proteins relate to histone post-translational modifications and how this in turn regulate transcription and how transcriptional programs then influence the developmental potential of human naive pluripotent stem cells. So to address these questions, we used a proteomics approach where we comprehensively determined the proteins that are enriched on chromatin in naive pluripotent stem cells and in prime pluripotent stem cells. So chromatin was isolated and then proteomics was applied. This led to a really exciting data set where we detected thousands of proteins and their abundance of chromatin. And we can now compare the abundance of chromatin proteins in the two different pluripotent stem cell states. And there we see a lot of changes, dramatic changes in the abundance of chromatin proteins. Of course, we see changes in the abundance of transcription factors, the key regulators of cell identity, and naive transcription factors are more enriched on naive chromatin, prime transcription factors are more enriched on prime pluripotent stem cells. And we also see a lot of changes in the abundance of chromatin regulators, including writers, readers, and erasers of chromatifications, as well as a plethora of chromatin remodelers. So we also compare these changes to mouse data, and we see that most of the changes we see here are concerned with mouses, but there are many exceptions as well. Many proteins behave in a human-specific manner. For example, DNMT3L is highly enriched in naive compared to prime chromatin. So we then wanted to understand the relationship between the abundance of chromatin modifications and histone post-translational modifications. So also using proteomics, we determined the abundance of histone modifications in naive and prime pluripotent stem cells. And there we saw that the histone H3 lysine 27 trimethylation, H3 kinase 7 trimethylation was highly enriched on chromatin in naive cells. And this was really surprising to us because we suspected that repressive chromatin modifications are associated with differentiation. In fact, John Gurdon had shown that as cells become increasingly differentiated in development, they become more and more resistant to being reprogrammed. And we had identified repressive chromatin factors such as the histone variant macro H2A that are associated with development and progressively help stabilize cellular identity and oppose changes in cell fate. However, what we see here is the opposite. We see that repressive histone modification, H3 kinase 7 trimethylation, is enriched in naive cells in the least differentiated cell type. So to gain more insights into the relationship between the abundance of chromatin factors and histone modifications, we integrated the chromatin proteome data with the histone modification data by linking writers and erasers of chromatin modification with the respective histone modifications. And in most cases, we saw similar trends between the modifiers and the marks, but not always. So we found that it's not always coupled. And for example, we found that for the H3 kinase 7 trimethylation, the writers of the mark is ZH2, parts of the Polycomb Repressive Complex too, and I will come to this in a minute. These writers of H3 kinase 7 don't change an abundance dramatically in chromatin, whereas the abundance of the mark does. So this suggests that it is the activity of the complex rather that changes rather between different cell states. So as I mentioned, H3 kinase 7 trimethylation is deposited by the Polycomb Repressive Complex 2, a developmentally regulated chromatin regulator that is evolutionary conserved. It's very important for regulating how gene regulatory programs are deployed in the embryo. And what we found using cut and run is that the H3 27 trimethylation mark is actually enriched at genes that are repressed and genes including trophoblast genes of alternative lineages in naive human pluripotent stem cells. So K27 trimethylation decorates trophoblast genes, which suggested to us that perhaps H3 K27 might restrict activation of trophoblast genes in naive pluripotent stem cells. So to test this idea, we found a very potent inhibitor of PRC2 that can inhibit PRC2 and completely deplete, or almost completely deplete H3 K27 trimethylation from the cells. But when we depleted H3 K27 trimethylation, not much happened. But when we induced differentiation, and this is work from Irene Talon, she found that inhibiting PRC2 during naive to trophoblast conversion promoted trophoblast induction. So this was very exciting because it implied that PRC2 may oppose trophoblast induction. And together with Nicolas Rivron, excitingly, in human blastoids, PRC2 inhibition promoted not only trophoblast fate specification, but also cavity formation. Again, suggesting that the activity of PRC2 opposes induction of alternative cell fates in human naive pluripotent stem cells. So in summary, we have comprehensively determined the chromatin proteome and abundance of histone post-translational modifications in human naive and prime pluripotent stem cells. These data are available as a resource for the community. You can browse the data online. We found that H3 kidney-7 trimethylation is more highly abundant in naive human pluripotent stem cells. And we found that PRC2 opposes naive to trophoblast induction. So altogether, these results suggest that naive human pluripotent stem cells are not completely epigenetically unrestricted as previously assumed, but instead use chromatin regulators to oppose induction of alternative lineages. And we propose that this is one of the ways by which naive cells in an embryo might stabilize naive pluripotency against weak pro-differentiation cues during development. So we knew that PRC2 is an important developmental regulator, but what we didn't know is that it acts this early in development, potentially as early as the first cell fate decision in an embryo. And this data is also an exciting starting point for further studying the role of chromatin factors in early human embryogenesis, because now we have candidate factors that we can now perturb and manipulate in the context of early human embryogenesis. And we are also pursuing these experiments not only in the context of stem cell embryo models, but also in human embryos directly. I now want to switch to the second part of this talk and tell you about a principle of chromatin-based dosage compensation in mammals. So during evolution, different sex-determining genes are acquired, and this leads to the acquisition of different sex chromosomes in male and females. And this happens independently multiple times in evolution. In the case of mammals, males have one X chromosome and one Y chromosome, and females have two X chromosomes. And so that creates a problem, a dosage problem. We heard beautiful talk from Sahin earlier on the gene sensitivity. But here we have a problem that we have two copies of the genes in males and only one in females, sorry, and only one in males, creating a problem for dosage sensitive genes. And so different species have evolved different mechanisms to deal with this problem. And one hypothesis in mammals is that the active X chromosome in males is upregulated to compensate gene dosage with autosomes. And this has been extremely well documented in Drosophila and C. elegans, but has been rather controversial in mammals. But I will show you later that it's definitely happening in mammals. Now, much more well established is the phenomenon of X chromosome inactivation in mammals to compensate gene dosage between males and females. And as a result, we think that the only active X chromosome in females must also be upregulated. So basically, Mary Lyon discovered and proposed in the 1960s that in early development in the embryo, one of the two X chromosomes is inactivated, that this is a random choice, so either the paternal or the maternal chromosome can be inactivated. It's induced by a long non-coding RNA exists that leads to inactivation of an entire chromosome. And it's been a really great paradigm for studying epigenetic regulation, the induction of chromatin silencing, and also memory of chromatin silencing, because the inactive X chromosome is a fantastic example of chromosome-wide memory of gene silencing that's inherited and maintained throughout a lifetime. And what I've always been fascinated by is that despite this extremely stable repressed chromatin state on the inactive X chromosome, it is reversible. It's reversed in the germline, but it can also be reversed by cellular reprogramming. And during iPS cell reprogramming, following the overexpression of transcription factors as discovered by Shinya Yamanaka, And during the induction of induced pluripotent stem cells by reprogramming, the inactive X chromosome is reactivated in a process called X chromosome reactivation. So I told you during development there is X inactivation. Well, one of the two X chromosomes inactivates and the other becomes upregulated. At least that's what we think is happening. And so we wanted to know, how is X chromosome dosage regulated during cell fate transition, and what is the interplay between X upregulation and X chromosome reactivation during reprogramming? So to address this question, we used transcriptomics, and we induced reprogramming of female fibroblasts with one active and one inactive X chromosome, induced reprogramming by transcription factor or expression, and then isolated fibroblast iPS cells, and then intermediate reprogramming, reprogramming intermediates, and then assessed gene expression using allele-resolution single-cell RNA-seq. And here we have a special way that can help us distinguish sequencing reads that come from either the maternal or paternal allele, so we can do allele-resolution analysis. So ordering the transcriptomes, we have a nice reprogramming progression. We can follow reactivation of pluripotency genes. We can see silencing of the X-cis-long non-coding RNA that needs to be repressed for X chromosome reactivation to take place. And we can also follow the process of X chromosome reactivation. Now we can look at allele resolution data for the X chromosome, and what we do is we take the sequencing reads from each X chromosome and take the ratio to autosomal genes. So it's the X chromosome to autosome gene expression ratio, but with allele resolution. So what you're seeing here is expression specifically from the inactive X chromosome in blue. So it's inactive to start with, as expected. And then during reprogramming, it's reactivated to autosomal level. And now, when we look at the active X chromosome in red, we see that actually there is upregulation. There is a global expression to higher levels than autosome, which is clear evidence for X chromosome upregulation in mammals. And moreover, during reprogramming, as the inactive X chromosome is reactivated, it appears that the active X chromosome reverses its upregulation. So it's as if cells sense that the inactive X chromosome is reactivated and then adapt gene dosage on the other allele accordingly. So to test this sensing hypothesis, we took advantage of the fact that female mouse iPS cells with two active X chromosomes have a tendency to lose one of the two X chromosomes, so to become XO. And so we can now test the effects of aneuploidy on a gene dosage on the X chromosome. So if we look at female iPS cells with two X chromosomes, each X chromosome is expressed to similar levels, but if one of the two X chromosomes is lost, either the paternal or the maternal X chromosome, then the remaining X chromosome is upregulated. So this is exciting data because it really suggests that mammalian cells have the capacity of sensing the number of active X chromosomes they have and adapt gene dosage accordingly. So how is this regulated? Well, we know that at least changes in transcriptional burst frequencies are involved. So one way you can increase gene expression is by increase the frequency with which productive transcription takes place. And we see that indeed the upregulated active X chromosome has increased burst frequency of transcription, and this is reversed during reprogramming. And then when one of the two X chromosome is lost, then upregulation and increase in burst frequency takes place. So we also were interested in chromatin, and so we used ATAC-seq in female fibroblasts, and there we found that the upregulated active X chromosome has enhanced chromatin accessibility globally compared to autosome. And so this suggests that the phenomena of X chromosome upregulation in mammals is involves changes in chromatin accessibility. We can also look at chromatin accessibility with allele resolution during reprogramming. So again, looking at the inactive X chromosome, it's inaccessible, condensed to start with, and then it reacquires normal accessibility level during reprogramming, whereas the upregulated active X chromosome has enhanced chromatin accessibility, is more accessible, and then loses this enhanced accessibility during reprogramming. So in summary, what I've told you is we have identified that mammalian cells have the intrinsic capacity of sensing and adapting X chromosome dosage. And this is really exciting to us. And it has recently been published in Genome Biology and confirmed by others as well. And so what is the significance? Well, we think that this helps to explain why X chromosome monosomies are only viable in humans. What by that I mean is that X chromosome monosomies is the only viable monosomy in humans. And so it could be that all other monosomies lead to lethality because they don't have a dosage compensation system that acts on a chromosome-wide level. But in the case of the X chromosome, the existence of a mammalian dosage compensation complex enables these patients to survive even despite the loss of one of the two X chromosomes. So, and then the future research is really, what is really exciting is that can we now identify the mammalian dosage compensation complex? And we don't think that it is actually fully conserved with other species because it evolved independently. So I don't want to tell you about a third really exciting piece of work where we have basically identified a way to model one of the missing human cell type. So when it comes to studying human post-implantation embryogenesis and what happens after the human embryo implanted into the uterus, there are many challenges. It's inaccessible for research. It's not fully conserved with vertebrates. It's poorly characterized. The morphology of the embryo is very different from rodents. And the current models we have do not fully recapitulate cellular diversity and morphogenesis. And what I mean is that there are even cell types for which no in vitro model is available. And what we discovered is that naive to trophoblast fate induction not only induces trophoblast stem cells, but also induces another unexpected cell type. And so we were puzzled for a while as to what these cells were, but by using single-cell transcriptomic and alignment to single-cell atlases of human embryos, we found that the unknown cells correspond to extraamniotic mesoderm cells. So the unknown cells express all the expected extraamniotic mesoderm genes from monkey and human data sets, but not genes from other lineages. So what is the extraamniotic mesoderm? But it turns out that the extraamniotic mesoderm is one of the key missing cell type. And by that I mean it's actually a really important cell type that arises a few days after the embryo implants. And then what you can see here at day 14 is you have the outer trophoblast cells. In the middle, you have the embryo with the epiblast in dark blue, amnion in light blue, primitive endoderm in yellow, and all these green cells here are the extraamniotic mesoderm. So the extraamniotic mesoderm basically connects the embryo to the future placenta. It forms the primitive umbilical cord, and it also forms the primitive blood in an embryo. So it's likely to be essential for human embryogenesis. However, very little is known. And what's fascinating is that it also occurs and arises in the embryo with species-specific kinetics. In mouse, extraamniotic mesoderm cells arise at gastrulation from the primitive strake. However, that's not the case in human. In humans, the cells arise in the embryo several days before gastrulation and from unknown origin. And as I mentioned, there were no in vitro model for human extraamniotic mesoderm cells. So I'm showing here an image of a monkey post-implantation embryo where you see the embryo in the middle with the epiblast in red, cytotrophoblast at the outside, and all these green cells here are extraamniotic mesoderm cells. And I hope you can appreciate how extensive this tissue here. It's a very abundant cell type in the early post-implantation primate embryo. But again, we could not until now make these cells in vitro. And what we found is that the cells we make in vitro actually do express the right genes, including VIM. And so we also determined, so we comprehensively characterize the cells, and then we have also determined the chromatin activity landscape using single-cell attack-seq. So we can predict enriched transcription factor motifs and predict a key regulator of cell identity that might drive the extraamniotic mesoderm program in the embryo. And we also identify candidate regulatory elements. And we know that disease variants are enriched at cis regulatory elements. And so we like to think that the cis regulatory elements that are active in extrahepatic mesenchymal cells might be enriched for disease variants related to infertility. So I want to summarize. I told you that we have found a model for the missing cell type that we think this could help in the future, and we hope that it could help understand important societal problems, because the cells really appear in the embryo at a time when many miscarriages and pregnancy losses take place. And so in the future, we think that we'll be able to use these cells to study the cellular and molecular mechanisms that underlie their emergence, and that might be misregulated in diseases, and also can tell us more about primate-specific developmental mechanisms, and of course, about how chromatin and epigenetic mechanism regulate early development. So I want to stop here and recap what I've told you. I told you that chromatin barriers oppose lineage induction in human naive pluripotent stem cells and blastoids. That naive cells are not epigenetically unrestricted. In the second part, I told you that mammalian cells have an intrinsic property of sensing and adapting X-chromosome dosage. And in the last part, I told you that human naive pluripotent stem cells can be used to model extrahepatic mesenchymal specification. And with that, I'd like to thank all of the people that have done the work. Key people in the work that I've talked about today have been Irene Talon, Dick Zellermans and Henrik Marx, Sigrid Verhelst, Martin Danins, Peter Ragan, Adam Bendal. And the work on X-chromosome dosage was by Adrian Januszewski and Irene Talon. And then the extrahepatic mesenchymal work was led by Dick Swanai-Pham, Amitesh Panda, and Bradley Ballatin. And I would like to thank you for your attention.